Hello, good morning everybody. So, we were talking about something where we can consider the modification of some electrodes. Why we require that? That is also important, and why we are talking all these things is basically important in that respect, that whether that developed electrode can be useful for sensing something. That means, for sensing purpose, whether we can use those electrodes such that, as we have seen, that something is reaching to the electrode in terms of the corresponding analyte. Okay. So, when analyte is reaching to the electrode, and depending upon its nature, whether it is a cathode or an anode, we see the oxidation and the reduction of the corresponding analyte species, and we can get the corresponding potential for that particular purpose and the number of electrons or the Coulomb charge which is passing through that particular electrode. Now, if the analyte is electroactive such that we can sense that particular analyte also, it can be your O2 or it can be simply a glucose molecule. That means, we have to go for, that means we can dump electron on it, okay, such that we can have O2 minus, we call it as dot, is a superoxide radical. So, one more electron if it gets, we get O2 2 minus. This we all know that how to convert O2 to superoxide to peroxide. So, this sensing business is very important in this way, that how we can sense, what you have to sense, because O2 is reaching, O2 is doing something, whether a water solution or aqueous solution or some industrial effluent has some dissolved oxygen. So, how we can measure the corresponding dissolved oxygen in terms of the electrochemical methods of analysis or the application of the whatever we are studying in this whole course. So, this particular one, that means the O2, if we are able to detect this one, or if we are able to detect this, or even we can detect the electron transfer reactions. So, these we should detect as the corresponding in terms of the electron transfer reaction. If it happens, if the electron transfer reaction is taking place on the electrode surface, we get that particular reaction. So, what do you see, that we are talking something, that we are talking something related to a gas species, a gaseous species. That means, O2 is going to the electrode, and we are thinking that O2 will get the electron from the electrode. So, you have the cathode, and cathode is reacting with O2, and you sense that particular one. So, this O2 is a gas molecule, then the glucose is a organic molecule, and then we can have proteins, etc. So, proteins can also accept or donate electron in terms of the corresponding attached electrode. So, if it is close by some electrode, this is electrode. So, these more and more complex species, starting from the gaseous species to that of your protein species, if it is nearby to that of the electrode, it is not that all electrode can sense the presence of these in terms of their electron transfer activity. So, we can go for something, that is why we require the corresponding modification of those electrodes, and the modification in terms of these basically give rise to something, that this size of these, that means the protein size is pretty big, and gas we know that something is that we can have some screen or the membrane which can permeate the gas molecules. So, because there are different sizes of these gases also, like O2, N2, and all their properties are also different. So, this thing, that means the modification in terms of the electrode. So, we can cover the electrode with some membrane, and if the membrane is gas permeable, So, if this membrane, what is enclosing your electrode, is gas permeable, and it only allows the O2 to reach the electrode. So, we get something that we can have the corresponding size is very important, and as a result we get some membrane. So, you have the membrane, you bring the membrane and related to size also. So, that is size exclusion. That means, basically what we do in terms of the filtration with a paper, filter paper, or a corresponding membrane type of thing, that is the cloth, the polypropylene cloth, we always know that, then also filter it out. So, this is some sort of filtration we can have, and the size exclusion membranes should be there. So, the size exclusion membranes will be there, and we will not allow in that case the direct contact. What we are looking for, direct contact of the electrode. So, we know that when we have the normal hydrogen electrode and hydrogen gas is bubbled nearby the electrode at a particular pressure, and we know that a special type of electrode is required to detect that particular presence of hydrogen and its electron transfer in terms of its electrode property as normal hydrogen electrode. So, this direct contact with the electrode, what we do, we go for something that is the size exclusion membrane we can put. So, we should go for something which is porous. So, a porous, again the porous membrane we should fix, and the species of our interest, that means whether it is a protein or a O2 molecule, that means the gas molecule. So, the species of our interest can only traverse. So, species of our interest can only traverse such that it can reach the corresponding electrode. So, when this is there, that means if something is there, that means if we think that is only permeable to gas, particularly the O2 molecule, the O2 gas molecule. So, if you have some mechanism where the porous membrane is attached, and that porous membrane can only allow your O2 molecule to reach the electrode, but it will not allow the bigger molecule having some kilodalton molecular weight. So, it is basically a big organic mass, polypeptide, several polypeptide chains are there, and that mass is also there. It can be 50 kilodalton or 80 kilodalton or 150 kilodalton of the mass. So, this protein will not go. So, protein is unable to do so. So, if you allow to think of the direct contact, so protein, the protein masses, basically the protein are unable to do so, that means to reach the electrode. So, the protein molecules will not reach the electrode. So, we must have some different mechanism. If we try to use the corresponding electron transfer reaction or electron transfer behavior for its potential as well as the rate of electron transfer for the protein molecule, we should have something different in mechanism. So, this is one, that means the porous part is there. So, now we can have that membrane. If we put, the second mechanism is the membrane, which is very important for your O2 determination. So, we put some membrane. So, membrane can separate those species. So, is a thin film of electrolyte can also be a membrane. So, if we have the membrane, so if we have the electrode and we say that we can have some porous membrane, but in between we can have a thin film of electrolyte. It has certain advantages. So, this particular one, if membrane can allow some species to go inside, then thin film of the electrolyte is another check. So, one particular such mechanism that we will study now, or that we will see how it is developed, how it is framed, is a, we call it as a oxygen electrode. That means, it can sense the oxygen, that means like your hydrogen electrodes is very easy to remember in that way. That is a normal hydrogen electrode. So, we have the oxygen electrode, and the person who introduced this having a surname is Clark. So, the scientist Clark invented that one. So, it is a Clark oxygen electrode. So, what it does basically, that means it first thing that what modification we make on the electrode and the electrode surface, such that some material like that of your O2 is our interest. So, this O2 molecule can reach. So, how you get that particular electrode, because we are talking in terms of this electrode. So, it allows the O2 to go, allows the O2 molecules rather to reach the electrode, O2 molecule to traverse, O2 molecule to pass. That means, it can reach the electrode by passing through this membrane. Okay. So, this particular one, so reach the electrode to traverse the membrane. So, it is passing through the membrane, and then what happens, that O2 can be dissolved in the thin film of this particular electrolyte. So, if you have the thin film, it is a very small film on this. We know that when we dip a solid electrode or a solid surface on some any liquid, so liquid can form a film on that particular solid surface or the solid support. So, your electrode is your solid support, and on that electrode if you have that film, and that film can dissolve this O2. So, O2 either dissolved or present in the gas phase. That if it is not dissolving in the thin film of electrolyte, it is present in the cavity and it is basically trapped. So, that is the corresponding mechanism, that how we develop and how we grow that particular electrode, which is your oxygen sensor. So, we get the different types of these oxygen sensors over there, and how we can develop those oxygen sensor. But the very first thing of that developing oxygen sensor is your developing the electrode. Otherwise, the mechanism for making your measurements for all these things, whatever you measure the current, because definitely the sensing mechanism is the corresponding amount of current what you can detect in terms of the corresponding oxygen present is it. So, it is required for why we require that oxygen sensor. So, it is basically the development of the electrode, and that electrode we can consider as a sensor. It can determine quantitatively and to a very low concentration level the dissolved oxygen in any solution. We are talking about that industrial effluent, the environmental material, the water from the sea, water from the river, water from the lake, anywhere. Because as we determine in terms of the corresponding talking in terms of the water pollution is a real headache for the chemist, is a headache for the environmentalist, and a headache for the other people also. So, we know two terms basically, we can the chemical oxygen demand and the biological oxygen demand, the COD and BOD values for these water molecules, because in that particular case if we talk in terms of the corresponding demand of oxygen, the one is the chemical oxygen demand and another is the biological oxygen demand. So, what we can think of that how much oxygen it can take by the species present in it, whether it is a chemical waste or a biological waste for any industry or any other activity, human activity, can give rise to that particular demand. But now the amount of O2, even in the pure sample of water, we do not know how much pure sample of water if you have and what amount of water you should have. So, if you have a very handy electrode type of thing or a sensing type of thing, you can determine the amount of oxygen what you require for the water, what you are putting for your aquarium also, because aquarium has some amount of dissolved oxygen, otherwise the species which is living over there, the aqua species, that means fishes and other things, will not survive. So, this is one aspect, and another is also is how much is there in our blood and any other body fluids. Because the dissolved oxygen in the blood is also important, is not that whatever amount of oxygen you have, which can be transported to the cells by your mechanism through hemoglobin and myoglobin. So, these are the oxygen binding molecules or oxygen binding metalloproteins, because you have the metal iron is there. So, these oxygen binding metalloproteins which are involved over there, it should get a particular amount of concentration of O2, because you look for some oxygen saturation with respect to the myoglobin and hemoglobin, and these two very important or interesting molecules are there. One is required for storing of oxygen, another is required for carrying the oxygen or transportation of oxygen for the regular use. That means what we use, that we require those oxygen for burning the food material. So, the basic formulation or the basic framing of these is very simple. Is one example from again Cook's book. In any other book also you can have the corresponding simple line drawing for this, but the basic thing what we are looking for, you have two end of the electrodes, and you see that the power requirement for this is also your one single dry cell battery power is 1.5 volt. So, these two ends, the positive end and the negative end. So, what you have? So, the negative end, that means the corresponding one which we can use as the corresponding cathode is ending at a platinum disc electrode. So, you have a platinum disc cathode. So, you can have that for hydrogen electrode also use platinum, the most possible inert metal ion which will not react the species either from the solution or the industrial effluent or the gases itself. So, basically this is the bed, I mean is basically disc, very small circular shaped disc is there, and disc is connected with this electrical connector to the negative end of the battery or the power source. And you have something that you have some insulating rod such that it is separated from your other material which is your anode material. So, you have the insulated rod, can be a graphite rod, can be any other carbon rod, can be other ebonite rod or anything. So, you have the insulating rod. So, insulating rod and the cathode is making, and you have the other one, that means the ring shaped anode. That means you have the thing that we will discuss very quickly, that you have this one is the disc. So, what you should know, because this has also the development of the type of the electrode. So, the mechanical framing, the mechanical framing of the electrode is very important. So, this the mechanical engineering can do. So, the mechanical thing is required for your fabrication, how you fabricate the electrode. So, this is your disc part, and outside you have the corresponding ring. So, this mechanism you should know that the inner one is your disc and the outer one is your ring. So, if this is your cathode, this is your anode, and you get the everything, that means your everything is enclosed in the corresponding inert material, and you have the connection. So, outside you have one connection, and inside also you have the other connection, because it is covering. So, once it is connected, it can get that thing, and inside you have the other connectivity. So, this is the thing that if you are asked to draw it also, you will be able to draw, and you should also be able to tell others that how it is framed. So, you have this, that means the platinum cathode. So, it is there as the disc, and surrounding this is your ring shaped anode, but this is made of silver, because you have the corresponding potential differences and all these things. So, this is therefore the electrode, the material what you are using, you should always be very much careful to know that particular material what you have, that electrode material. So, at one point, because it is a very small amount, only very little amount of platinum and a little amount of silver is required. So, that is why this is not so costly if you compare the corresponding labor charge for making or fabricating the particular electrode. So, material wise it is not much costly, but the fabrication wise it should be costly enough. So, you have the silver electrode, then what you put? You put a buffered KCl medium. That means what we know, that when we write any cell or half cell, you know that the KCl is there and silver is present. So, this thing is also there, so buffered KCl solution is there. And now what we are talking just now, that you can have a corresponding thing, that means two things are there. The first one is your membrane, which is outside your electrode. So, membrane, so it can be replaceable also, because after some use, after several experiments, you replace that. It is of only 10 micrometer thick and which can permeate your O2. So, if the thickness of that membrane, whether it is a polypropylene cloth, we consider this as a polyethylene cloth, the polypropylene, the PP cloth, or any other material of that thickness, but it should allow oxygen to pass. And you have another very thin layer of KCl solution. So, it is buffered in KCl, and if the layer is there, that means what we just now have seen, that you have a corresponding separation, that means the thin layer of some solution, thin layer of liquid, and that is of that particular, and it is of the same thickness in the range of 10 micrometer thickness. So, what you get? You get a porous membrane, and covering that particular one is a corresponding liquid membrane. So, this is a very simple fabrication for that electrode, is simply that you dip the thing and you measure the corresponding current due to the reaction of this O2 reaching the cathodes. So, a cathode platinum disc you have, which is your WE, is your working electrode, and embedded in an insulating rod, just now what we discussed. So, you can have that thing in writing also, that you should keep in your mind, and which surrounds basically a lower end is a ring shaped silver anode. So, is a ring. So, you have the disc and you have the ring. So, is a silver. So, you get this particular thing, that a particular arrangement which is known as your ring disc or disc ring electrode. So, the insulator through which it goes kept inside the second cylinder and containing buffered solution of KCl. So, the tubular insulator and electrodes. So, a insulator is there and electrodes are there, which is kept again inside a KCl solution, and sometimes 10 to 20 micrometer of permeable membrane. Now, you see, like polypropylene, you can also use a Teflon. So, Teflon we all know is a homemade, now there is a use in the home maximum. So, the Teflon coating on the kitchenwares and all this in kitchen, you get the Teflon. So, it is a very useful material. So, is basically a membrane of Teflon is placed at the bottom of the tube by a O-ring, and thickness of the electrolyte solution between the cathode and the membrane is again only 10 millimeter. So, this 10 millimeter to 10 micrometer thickness of these we can handle for reaching that particular oxygen for a typical diffusion. So, adjacent to the disc cathode and then diffuses the electrode for reduction to water. So, what we are looking for? We are looking for the reduction of O2 to water. So, that is the only reaction what we are looking for, as I told you. So, we are not going to those processes, that means we are looking for reduction of O2 to superoxide to peroxide. So, we are not going for that. So, anything can also happen over there, that means the fate of the oxygen. In one way it can go to superoxide to peroxide, and in other way it can go to formation of water molecule having a different electrode potential for the formation of water molecule from your O2 molecule. So, two diffusion processes are therefore are involved. They are taken part actively for this measurement process, one through the membrane and other through the solution between the membrane and the electrode surface. So, first it goes through the membrane, as I told you that it is passing through the membrane. So, it is crossing your membrane. So, this membrane it is crossing. So, it is also a diffusion process, and between the membrane and electrode surface you have the thin layer of that electrolyte, it should also pass through that. So, if the traverse of those molecules, that oxygen molecules, is diffusion controlled, so it should have two barrier, one is the typical membrane and another is your thin film of electrolyte. So, you get two different diffusion processes. So, this particular thing, that means the flow of those gaseous molecules, we can consider it as the hydrodynamic flow of stirred solution if we have. So, if stir the thing, so what we basically get that thing. So, is a in aqueous medium and you just stir the solution, is the dynamics is there. So, hydrodynamic flow of stirred solution is based on measurements made with a rotating disc electrode. If we can have, so the mechanism is also there. So, what you know that now we are bringing something which is little bit complex, not very much complex, because you have the disc, you have the ring. Now, you do something again, something where we have the stirring thing, that means we rotate, we can rotate now. So, is the rotating of that particular ring. So, you can rotate the ring nicely. So, you have the corresponding rotating disc electrode. So, with a rotating disc electrode, so the ring can be rotated and the disc also can be rotated. So, what you rotate? So, this is a very complex process related to your diffusion and the hydrodynamic flow of the analyte. So, you can have the option that you can rotate the disc as well as you can rotate the ring. So, one electrode what we will find, and we can only learn also that if we abbreviate is as RDE, the rotating disc is there. So, you are not rotating the ring. So, rotating disc electrode. So, this is one type of electrode that we can have of the same formulation or same mechanism what you get. So, you get that. So, you have just now what I have drawn, you can also get this from any book, and you can also draw yourself that what does it mean, what is the thing that if you given the diagram also level it. So, it is in terms of this particular classes, that means electrochemical classes, where we try to understand everything in terms of the corresponding thing, and this is basically a very simple type of electrode. That what are the shapes of those electrodes, we should always know that when you have a hanging drop of mercury. So, drop of mercury, so hanging drop of mercury can give rise to electrode, and what is the shape? That shape is known, that is have the thing that through which a capillary is there. So, is a very fine capillary is there, and you get a corresponding drop forming, because depending upon the size of this capillary, this corresponding internal diameter, you get that corresponding size of this mercury drop. So, we get dropping mercury electrode, and this dropping mercury electrode is that through capillary you are going for this drop, and for some time that it has some lifespan when it is hanging. So, life when it is hanging from the bottom of this capillary or the corresponding solid surface of that electrode. So, this dropping mercury electrode you can have. So, you should be able to draw the shape, and also the ring shaped or the disc shaped thing is also there. So, if you now the rotating ring disc electrode is a modified rotating disc electrode. So, rotating ring disc electrode. So, disc is there and ring is there, as we know now. So, if you have ring as well as disc, now the rotating disc electrode is useful for studying different types of electrode reactions. So, now you have the disc and you have the ring surrounding this particular disc, and everything you have this particular one. So, you can have the option that you can rotate the disc, you can also rotate the ring. So, when an electroactive species is generated at the disc, so disc is basically generating that electroactive species. That means what you have, that means if the disc is taking that or supplying that electron for your reduction to your O2 species, it is forming your O2 minus, or you can go to O2 minus, that means the oxide ion. But in this particular case, when it is rotating something, that it swept passes the ring where it undergoes a second electrochemical reaction. That means it is passing from here and going or reaching to the ring, and there it goes and taking some other reaction, because it is a electrode of different polarity. If the disc is cathode, your ring is anode. So, after getting that thing, it is reaching the anode. So, you get something which is different, but what you are looking for is basically a cyclic process, the way we have understanding the thing in a cyclic voltammetric measurement, that you do first oxidation, then you go for the reduction. Here also the disc is doing something, that means if it goes for reduction in terms of a cathode present in the connection for its disc, when it reaching the ring can go for its corresponding oxidation. So, all these studies provide very useful informations about the mechanisms and the intermediates in the electrochemical reaction. So, this will be a much more complicated process, but you should know a little bit only that the modification of those electrodes can provide more and more useful information in terms of the corresponding thermodynamically controlled electron transfer or thermodynamically controlled the corresponding kinetic behavior. So, what we find that is that for important that when we go for the corresponding kinetics and all these measurements. So, if we have these two, that means if we have the disc as well as the ring in hand, so we can have two different currents in our hand. So, separately we can measure, because we can measure, as we know that working electrode is something which is your cathode, working electrode can be your anode also. So, you have a disc, A is the disc current and B is the ring current what we get. So, for reduction of oxygen at the rotating ring disc electrode. So, is the rotating ring disc electrode. So, we can also consider the rotating disc electrode or rotating ring disc electrode we can consider. So, what we do, we just basically plot, again it is the current potential plot what we studied earlier, that now the potential of the disc is plotted against the current of the disc, and in other case the potential of the disc is plotted against the current of the ring. That is the only difference. So, these two are different, the shape of these plots will also be different, but the basic reactions for this is all same, that is the electron acceptance by the O2 molecule for its reduction, and your corresponding O2 is also reduced for your hydroxide ion formation as well as the water formation. So, here also this thing is also the reverse reaction what we get, that is your H2O2 plus 2OH minus what is on the right hand side what is forming, because this has been formed, that means hydrogen peroxide has been formed by the disc. So, it will be reaching to the ring. So, ring current is due to the again that corresponding reaction of hydrogen peroxide, that means the oxidation of the hydrogen peroxide. So, you see that this particular reaction, that means this current is, you have a plateau and then again it is falling depending upon the concentration of the H2O2 what is reaching over there. So, the corresponding sense of the ring current is completely different. So, these two plots basically will be useful when we talk in terms of the corresponding development of the electrodes. Thank you very much.